So good afternoon, everyone. This, I hope, will just be a short class just to end our discussion about genes to proteins, about how DNA actually drives the whole functioning of the cell by directing the synthesis of the enzymes, proteins, everything else that is required by the cell. So one of the things that we need to think about is that although DNA is replicated really accurately every time the cell divides, if that replication was 100% accurate all the time, every single time a cell divided, then we would all still be bacteria swimming in the primordial seas, because it is essential that DNA be able to change for evolution to occur. So evolution is the result of the accumulation of slow changes in the genome. And those changes to the sequence of DNA in the genome occur because mutations occur. That is, changes to the sequence occur. Can be for various reasons. It can be spontaneous. It can be an error in replication. It can be because the DNA is damaged and then is then repaired, and it's not repaired exactly the same as it was originally. There are all sorts of reasons why a DNA sequence may change. Any change to the DNA sequence is likely to have an impact on the functioning of the cell. Very often, changes in DNA sequence by mutation are deleterious. And the deleterious mutations are very often lost quite quickly from the population. But sometimes a mutation can occur which is beneficial. And when that organism that inherits a beneficial mutation operates, it operates slightly better, it reproduces slightly better, and it hands on that improved DNA sequence, if you like, the changed DNA sequence to its progeny. That is the stuff of evolution. That is how evolution works. So we need, before we finish our discussion here about the functioning of DNA, we need to understand just basics about mutation, and what the impacts of mutation are. So mutations, by definition, are changes in the genetic information contained in the cell. They are changes in the DNA sequence. We are going to mostly think about what we refer to as point mutations. And that is, these are mutations which are changes where one base pair is altered. Remember that the base pairs have to match. So usually a mutation occurs in both bases, in both bases which are paired to one another across the DNA spiral. But even a single base pair change can have drastic effects on the functioning of the organism, because it can change the codon, which results when mRNA is produced, and it changes the amino acid which is put into the protein, for example. So it could actually have drastic effects on the functioning of the protein, and especially on the functioning of enzymes, whose function is intimately tied to that DNA, to the sequence of amino acids in the polypeptide that make them up. So let's have a look. We've mentioned this one before, but it really serves to illustrate the impact of a single base change that a single base change can have. And this is the mutation which results in the condition of sickle cell anemia. So sickle cell anemia arises in one of the hemoglobin molecules. There are 4 hemoglobin molecules, 4 peptides that make up the hemoglobin molecule. And in one of them is basic globin, and here is the sequence that we're particularly interested in. It's 3' CTC, 5' GAG, there it is there. And here is the mRNA, which results GAG from the gene. So there it is there, and that normally codes for this glutamine. In sickle cell hemoglobin, however, there is a mutant. And there's a substitution, not CTC, but CAC, and the resulting mRNA will be GUG. And that is for valine, a completely different amino acid. And that has huge impact on the functioning of the hemoglobin molecule. Normally, hemoglobin picks up oxygen in the lungs in an oxygen-rich environment, and it's bound quite tightly to the hemoglobin. It's bound until such time as the hemoglobin reaches an area of low oxygen tension, like in the periphery or in the cells, wherever, at which point the hemoglobin liberates the oxygen. There are changes in shape of the hemoglobin molecules as this happens, but they remain flexible. And the whole red blood corpuscle is actually able to deform as it makes its way through the very, very fine capillaries back into the venous system to go back up to the heart. In sickle cell hemoglobin, that is not the case. Instead, under low oxygen conditions, when it liberates the oxygen, drops its oxygen, the hemoglobin molecules actually form fibrils, and they don't return to a normal shape. And they pull the blood corpuscle into the sickle cell like this. And the sickle cell is brittle. It can't make its way through the capillaries easily, and they tend to clog up the capillaries. They set up an inflammatory condition in the capillaries. And very often this can have very, very bad impact on the organs and areas of the body where these capillaries get clogged up with these sickle cell blood corpuscles. So this single base change is actually a base pair change, don't forget, TA to AT. It's quite easy to do. All you've got to do is flip it, right? And that is something which can happen quite easily. Ends up producing this completely different codon, a completely new, different amino acid inserted where this should have been. So that is an example of a single nucleotide being changed, a single nucleotide pair substitution. We're going to see that there actually this can have various impacts. And there is one other kind of mutation that we're going to consider, and that is where we actually insert an extra base pair. So all of these are going to be, we're going to see, first of all, what happens with substitutions of bases one for the other. We've seen one in sickle cell. We'll see some more now. And first of all, we sometimes see that a mutation, actually quite often, mutations in the DNA have no impact whatsoever. How can that be? Well, it relies on the fact that the DNA code is redundant. That is, there for one amino acid, there can be several different codons. We saw that on our table of codons, right? So let's have a look here. Here is our wild type DNA. Here it's coding for glycine. And here's the template strand here, CCG, and the messenger RNA, the codon that would produce GGC. Just remember that we could actually have several different codons coding for glycine, but all of them had this pair of bases, GG. Then there was a third. It could be C, it could be G, there could be various things there at the third base of the codon, and it made no difference. It's still coded for glycine. That economizes on the number of tRNAs, because any tRNA with an anticodon CC, whatever it is, can fit here. We call that wobble. That the tRNA could actually, one tRNA could fit on several different codons. This has an important effect, because if I introduce a mutation here at this third position of the triplet, I've actually got quite a lot of latitude. Have a look here, CCA instead of G. So that adenine instead of guanine, there you've now got CCA, but it still codes for glycine. Even though that's been changed, it still codes for glycine. This we call a silent mutation, because it's not going to have any impact on the phenotype at all. It is a mutation, it's a change in the DNA sequence, but it has no impact. We regard it as being silent. So this is not always the case, as we've already seen with sickle cell anemia. If we change one of these base pairs, we can actually change the codons sufficiently that we bring in a new amino acid. And have a look here. We're using the same triplet again. CCG here on the template strand. Normally it would code for GGC, as we saw with glycine. But here in this mutation, we substitute, instead of C, we have here a T. And we now have TCG, the mRNA, AGC, that codes for serine. A completely different amino acid. We don't know, I don't know what effect this would have here, but we saw before that that single change to valine in the hemoglobin resulted in the hemoglobin phenotype. So it could be really serious. On the other hand, we don't know for this. This could actually be beneficial. If this was a beneficial mutation, then it could become fixed in the population and end up contributing to the evolution of this enzyme and the evolution of the whole of the organism itself. So this is a missense. It is a base change which results in inserting a completely different amino acid to the original. Now, remember that there are 3 codons which actually code for stop and instruct the ribosome to stop translation entirely. And it is possible to actually change a codon in the middle of a sequence, which should be leading to production of a polypeptide, to alter one of those base pairs, and it results in a stop codon. So have a look here. It's a kind of a missense in a way, because we're going to change the codon that results. So here again, you've got glutamine here, CAG, and we're going to substitute a T for that C on the template strand, and have a look and see now what mRNA you're going to produce from this. We're going to produce a stop codon there in messenger RNA. And if you introduce a stop codon, then translation is going to end. And instead of producing this long string of amino acids here, we're going to end up producing a shortened, truncated amino acid, truncated polypeptide rather, which quite probably is not going to work. And this is called a nonsense mutation. It is when you end up substituting one of the stop codons for the normal codon, that should have been used. The next one are not these point mutations. They're not single base changes. All of those point mutations did not alter the total number of bases that were in a particular gene. But now we're going to have a look and see what happens if we insert or remove bases and change the number of base pairs that are present in a gene. And these are called insertions or deletions. Sometimes you'll see that if you go on to study any molecular biology, you'll hear them referred to as indels, insertions and deletions. And they're additions or losses of nucleotide pairs. And these are often extremely deleterious, and for a really important reason. And that is that they introduce what is referred to as a frame shift. Remember that the ribosome, for example, needs to know exactly where to start. It begins at an AUG, at a methionine, reads it AUG, then the next codon, which is 3 bases, then the next codon, which is 3 bases. If we do anything which alters that frame of reference, then the ribosome may carry on translating, but will translate something completely different to what the original sequence was. So let's have a look here, and you'll see. Here is the normal sequence, CAG, here CCC, that's the second codon, ACT is the third codon. And this is, here are the amino acids that is coded for glycine, proline, threonine. Here is our first examination. What happens if we actually insert something that doesn't belong? And here we have CAG, first codon, just as normal. Now we're going to insert a T, a thymine there. Now the second codon reads TCC. What does our third codon read? CAC. It's been shifted. The frame has been shifted. There it is there. Now it's here, CAC, a new codon. And in fact, what happens now is that downstream of this insertion, we change the whole sequence. Now instead of proline, threonine, we have serine, histidine, completely different. It's a completely different sequence. Who knows what the impact will be, but it's surely not going to result in anything which functions like the original polypeptide. So that a similar thing happens if we insert 2. We shift the frame 2 bases and still end up with a completely different sequence. Notice though what happens if we insert 3. If we insert an entire triplet in the original DNA sequence, if we insert an entire triplet, we will insert an entirely new codon. Here it is here, phenylalanine. Now have a look and you'll see, here's our original ding, ding, ding. Here it is here, ding, ding, ding. There's the original, but we've inserted here a new codon and hence a new amino acid. This may itself have a tremendous impact on the functioning of this polypeptide, but at least the frame has been restored for everything downstream because we inserted 3. But we've inserted a new amino acid. These are insertions, but a similar thing happens if I take out a base. If I take out a base pair, if I delete a base pair, if I delete 1, I introduce this a frame shift. My codons would then read CCA here instead of CCC. It would read CCA. Similarly, if I took out 2, I would shift it back to CAC. It would be reading, the frame would shift. But if I took out 3, then what I would do is I would simply be removing an entire codon. I would, let's say I'm pointing to this one here. If I deleted these 3 here, I would delete proline from the sequence. Could have very serious effects, but often that is a lesser effect than the frame shift of 1 or 2. So those indels, the insertions and deletions, often extremely, extremely deleterious because they have the capability of changing the entire sequence of amino acids that are put together. So these mutations can occur spontaneously during the normal functioning of the cell. Errors do occur during replication of the DNA, especially. The DNA polymerases can sometimes put in the wrong bases. As we heard before, there are DNA repair mechanisms, but the difficulty for the DNA is often, for the repair mechanism, often to tell which is the correct sequence. For example, most of them depend on the fact that bases are supposed to be paired. So if I, for instance, I had an AT pair in my DNA, and instead of inserting AT, I inserted C. So I had AC. The cell may know that something's wrong because A and C are not supposed to be together. They're not going to bond properly across the width of the DNA strand. But it's difficult for the cell mechanisms to know which is correct. Is it A, which is correctly inserted and must have a T, or is it C, which is correctly inserted and must have a G? And sometimes it'll make the wrong choice on that, so that we can end up with a new base pair being inserted. So there are all sorts of cellular mechanisms for trying to get around this, but they don't all work as well all the time. So we do definitely accumulate mutations in our DNA, even as a normal part of our functioning. But remember, the important mutations are those mutations which occur in the germ cells, in those cells which are going to produce gametes. We may have changes in our somatic cells. We may have mutations in our somatic cells, for example, that are very deleterious to us because they set off tumor processes. But those kinds of mutations pertain to the organism itself, not to its progeny, but it is mutations in germ cells that are particularly important. Anything which will be handed on to the progeny is of particular relevance, especially in terms of evolution. There are various ways in which the DNA sequence of cells can be changed quite apart from the normal processes of replication. And one of them is that we are subject to the action of mutagens, of substances which will induce change in DNA, often by damaging the DNA, and then repair mechanisms repair imperfectly and insert new DNA sequence. There are many of these. If you want to, I'm not going to discuss them in detail here, just to remind you that we do take in mutagens the whole time. We are just naturally, we are exposed to mutagens. But unfortunately, also in the industrial era, we are subject to many new substances, synthetic substances, this sort of thing, which are mutagenic, which can actually alter DNA. And if they alter somatic DNA, they can have the effect of, for example, sparking cancers, that sort of thing. But if they operate on a germ level, then they have the capability of inducing change in progeny. So just very briefly to segue to something quite different, and that is that we look at many of these genetic diseases, such as sickle cell anemia, and we realize that they have resulted from relatively small changes in DNA sequence. And of course, we have always been very interested in curing these diseases and finding some way of dealing with them. In the case of things like sickle cell anemia, the current kind of top-line treatments would be to try to culture stem cells, for example, in the blood, which do not have that mutation, and encourage their growth, and then perhaps do blood bone marrow transplant in order to replace the faulty cells. That would be the kind of current line of treatment for somebody severely ill with something like sickle cell anemia or even a leukemia or something like that. But in the last 10 years or so, an entirely new system has been devised, which is called a gene editing system. And this is a way for us to actually address particular DNA sequence. So that if, for instance, we know that in the sickle cell, that single base pair alteration has taken place, is there a way for us to go to the DNA itself, take out that faulty base, and replace it? And up till now, the answer has been no. But in bacteria, a system is present which recognizes specific DNA sequence and cuts at that particular sequence. We've already heard one example of this, and that is the use of restriction enzymes. Restriction enzymes actually cut DNA at a particular sequence. The restriction enzymes are a group of enzymes present in prokaryotes, and their real aim is to protect the bacterial cell from infection by bacterial viruses or phages. Many of those bacterial viruses have DNA as their hereditary material. They inject the DNA into the bacterium, it takes over the bacterial machinery, and it turns the bacterium into a viral factory. But bacteria can fight back. They have enzymes which specifically cut up viral DNA into short pieces and inactivate it before the virus can take hold. It turns out that the bacteria actually keep pieces of that viral DNA in a sort of library. And it's recognizable in the bacterial genome in an area which is called CRISPR. And that stands for clustered, regularly interspersed, short palindromic repeats. Right? Clustered, regularly interspersed, short palindromic repeats. It looks like an area where there are many very short little sequences, all the same, which when you look at the bacterial genome, they read the same forward and backwards on the two strands. And it's like hyphens through the bacterial genome. And in between them, the bacteria insert the fragments of viral DNA, and they're keeping it there as a library, as a memory. And if they are again infected by viruses, then they have an enzyme called Cas9, which will go to that library, and it will read the library, and it'll get information about possible viral DNAs which are coming in. And it will read old viral DNA sequence, and go and look for it. And if it finds it, it will cut that. It will cut it to pieces. So this is a system which works. We have this protein, Cas9. It goes to the library, and it makes a messenger RNA strand corresponding to a length of previously seen viral DNA. So that is called the guide RNA. So let me just go to the diagram. It's easier to explain. Here's the Cas9 protein there. It goes to the library and it makes a complementary RNA sequence. This is complementary to an old viral gene, piece of an old viral genome that the bacterium has seen before. And it forms a guide for the Cas9. The Cas9 in a bacterium, that Cas9 can wander around the cytoplasm, matching this RNA to any DNA it sees. And if it matches this RNA to a DNA, probably viral DNA, it will match it and then it will cut the DNA there and inactivate it. So scientists realize this gives us a way of editing our own genome, of editing a genome now, not in the prokaryote, but in the eukaryotes. And here's what they do. They want the Cas9 to go and cut a genome at a particular place. They make an artificial RNA strand. They take the Cas9 protein, they make an artificial strand. We know the sequence we want to cut at, the DNA, put the two together, insert it into a cell, and let it just wander around the cell until it finds the matching DNA. And it will then cut it. This gives us two possibilities. The first is that we could put Cas9 into a cell with a malfunctioning gene and cut that gene and inactivate it completely. There's one disease where this is a real hope, and that is the disease Huntington's disease, which produces these kind of strange proteins, which result in the illness. And this is one way it could possibly be treated, would be to, if we could get in and cut that gene and inactivate it. The second thing that can be done is, as soon as a eukaryote cell sees damaged DNA like this, DNA which has been cut, then it will attempt to repair it. And it'll either just stick it together like this, or else it'll try and insert a piece of DNA. And this is the other great hope of this system, is if we provide lengths of DNA in the environment where this cut is being made, then the cell will insert those into the DNA, and we can insert new DNA sequence. We can do it for one base pair, or we can do it for more base pairs. And experimentally, this works extremely well, and it gives us the hope, for example, that if you know that a couple who are having a child are likely to have a child with a particular genetic defect, that the gametes of that pair or the zygote, a newly fertilized egg, could be treated with Cas9 CRISPR, and the gene could be altered. In addition, if need be, for crops or whatever, we could actually begin to design genes where we inserted particular information that we wanted. So what I want you to do is I want you to have a look at this movie here. This is the discoverer of this CRISPR system, one of the discoverers of the CRISPR system, and who's this year won the Nobel Prize for her work. And she explains this there. The reason I want you to, in particular, just get a clearer idea of how the system works, but also to begin to think about some of the ethical problems. Because we're beginning to now, this system is really, in an established laboratory, quite an easy thing to do. And this opens up the possibility, which has already been exploited unethically by some scientists, of altering genes so that they look like we want them to look. Now, when we talk about disease, we think that that is fine. But what about if we want our children to be blonde rather than brown haired? What if we want our children to be tall rather than short, etc.? These systems open up that possibility, and we have to think very carefully about the ethics of that kind of work. So the CRISPR-Cas9 offers tremendous amount of hope, though, for genetic disease in particular. And sickle cell is right at the top of the list, right at the top of the list for being one of the diseases which might be helped. Because, for example, in sickle cell, there is a possibility that we could extract blood stem cells from somebody who suffers from the disease, culture their blood stem cells, alter their genetics, and then reinsert them into the person in the hopes that they would grow and then produce normal hemoglobin. That would be one strategy. There's other that I mentioned. Huntington's is another one. Cystic fibrosis is another disease where they're looking very carefully to see if CRISPR-Cas9 could actually be used. So we come to the end of this discussion about the DNA molecule and how the DNA molecule is worked. Keep in mind our attitudes towards this continually change. Remember how Mendel viewed what he was seeing in his pea plants and things like that, as he understood that he was watching the passage of discrete units of inheritance. He had no idea about DNA, no idea about the functioning of the cell. He didn't even know about how cell division worked or what chromosomes were. Then our ideas changed, and we realized that a gene is in fact a specific nucleotide sequence on a chromosome, that each of our chromosomes is composed of a single very long length of DNA, and each of the genes is a particular region of that with a particular sequence. And the importance of that sequence was that it codes for a specific polypeptide chain. And that polypeptide chain acquires its characteristics by its amino acid sequence. So we could then come to this definition. It's a region of DNA which can be expressed to produce a final functional product. Remember, though, that DNA codes for two things. It codes first, ultimately for protein, but second of all, it also codes for all the functional RNAs in the cell. It codes for the tRNAs. It codes for all of the small RNAs which splice together. The messenger RNA, for example, in eukaryotes. Many other codes for ribosomal RNA. There are many, many functional and structural ribosomal molecules. RNA molecules, which DNA codes for, in addition to polypeptides. Okay, so I'm going to leave it there for the moment, and then we move on our next classes to talking about evolution itself and how the processes of evolution work.